Hello, everyone. My name is Hanna Holmes, and today I will be introducing trehalose dihydrate. My principal focus is in protein engineering and biologics, and trehalose dihydrate will be the central topic for my presentation today with respect to its role as an essential molecule for biologic manufacturing and administration. This is possible due to its wide range of unique material properties, particularly with respect to thermal properties. Firstly, I will cover some background information on what a biologic is. As the name suggests, biologics are a class of therapies derived from biological origins, such as proteins and genes grown from cell cultures. This includes a wide range of different applications, including vaccines, monoclonal antibodies, and immune modulators. A specific example of this is the monoclonal antibody known as Herceptin, active ingredient named trastuzumab, which is an intravenous cancer treatment for HER2-positive breast cancer. Oftentimes, in these treatments, the active ingredient is considered to be the most important component in its composition. However, in order for these biological ingredients to be able to be stored and safely administered to patients, other inactive ingredients known as excipients are required. Excipients serve as vehicles for the active ingredient in improving the stability and enhancing the properties of the drug. In this example, Herceptin is formulated alongside excipients such as histidine, polysorbate 20, and trehalose dihydrate. Excipients are involved in almost every drug class, irrespective of the method of administration, in order to allow for the drug to be stored at freezing temperatures for extended periods of time and still be able to retain its effectiveness and structure, among other things. This is important because extreme temperatures, varying pHs, and hostile conditions inside of the body, especially with sensitive active ingredients such as proteins, which are prone to denaturation, would otherwise make delivery impossible. For injectables specifically, since they enter the body directly, either through the bloodstream in the case of intravenous injection, into the muscle and intramuscular injections, or under the skin in subcutaneous injection, these therapies require extra levels of sterility and chemical stability versus, for example, an oral route drug. Excipients for injections typically include pH regulators, isotonic regulators, antibacterial agents, and antioxidants. What makes trehalose dihydrate unique? Trehalose dihydrate, which is a crystalline, non-reducing disaccharide, is a chemical chaperone for trastuzumab as well as a wide range of other applications such as food products, vaccines, and cosmetics. Trehalose, the anhydrous form of trehalose dihydrate, is a naturally occurring sugar that is found in bacteria, fungi, plants, and some invertebrate, and are believed to be involved in supporting organisms against the effects of dehydration and freezing temperatures. Trehalose dihydrate is usually acquired by drug manufacturers as a white powder, as shown to the right, which is mixed with the active ingredient prior to storage. Trehalose dihydrate is used widely as a polymer due to its unique properties of enhancing cryoprotectivity, anti-aggregation, structural stability, and low reactivity. Trehalose dihydrate's intrinsic stability is imparted onto it by its structure. As I mentioned in the previous slide, trehalose is a non-reducing saccharide, which means that the monomers are linked at the anomeric position and cannot break down into a smaller form. This can be seen in the diagram to the right of trehalose, highlighting its rigid alpha 1,1 glycosidic bond. This also imparts interesting thermal properties that will allow it to resist deformation under extreme temperatures, with glass transition state between 110 degrees Celsius and 120 degrees Celsius, which is the highest among disaccharide polymer materials. The glass transition state represents where polymers transition from a glassy crystalline state into a malleable state. Therefore, at temperatures under that threshold, proteins are encased in a glassy protective coating to prevent denaturation at both high temperatures and preventing ice crystal formation and aggregation during freeze drying, also known as lyophilization, due to a complex series of multi-step thermal dehydration, in which the crystals change shape and structure before reaching the anhydrous amorphous form, shown in the graphic on the right. Furthermore, it prevents dehydration of these molecules as the crystalline form contains water. This keeps the active ingredients stable and protect post-thaw so that they can continue to work. Here's a 3D model that I created in the visualization software Avogadro of trehalose dihydrate. First, I constructed the trehalose, which is the large molecule you can see in the center, with the two conjoined ring structures, and then placed two water molecules in complex with it, attached via a hydrogen bond with one of the hydroxyl groups. Since trehalose dihydrate occurs as a crystal, I was able to find literature values from a study that used direct methods to determine the unit cell dimensions and space group of trehalose dihydrate, and used this model in Vesta to create a model of what the crystal structure looks like. Partially due to the highly complex shape and strong hydrogen bonds within the structure, a unit cell falls under the classification of the triclinic structure, which means that the sides are unequal and the angle of the unit cells are not necessarily perpendicular. In the study, the measurements 7.59 times 12.23 times 17.88 angstroms were used with 90-degree angles for simplicity. Using the found symmetry of space group 19, the molecules are sorted through the unit cell. This is the same model, but with the space filling model of the molecules rather than the ball and stick model in order to get a better visualization of how much space each of the atoms take in their electron clouds, and shows a fuller representation of the crystal structure of this unique molecule. In conclusion, trehalose dihydrate is a versatile and cornerstone excipient for biologic and pharmacological formulations due to its stability and structure, which provides unique thermal properties. It is a naturally occurring crystalline polymer that is notable for mostly its cryoprotectant, anti-aggregation, and non-reducing capabilities, and is used in many applications across the pharmacological industry and beyond. For those interested in learning more about this intriguing molecule, here are resources that made this presentation possible. Thank you for joining me and have a great rest of your day.